The big idea. Neuroplasticity, the ability of neurons to change their structure and function in response to experiences, can be turned off and on by the cells that surround neurons in the brain, according to a new study on fruit flies that I co-authored. As fruit fly larvae age, their neurons shift from a highly adaptable state to a stable state and lose their ability to change. During this process, support cells in the brain, called astrocytes, envelop the parts of the neurons that send and receive electrical information. When my team removed the astrocytes, the neurons in the fruit fly larva remained plastic longer, hinting that somehow astrocytes suppress a neuron's ability to change. We then discovered two specific proteins that regulate neuroplasticity. As fruit flies develop, special cells surround their neurons and seem to halt neuroplasticity. Why it matters. The human brain is made up of billions of neurons that form complex connections with one another. Flexibility at these connections is a major driver of learning and memory, but things can go wrong if it isn't tightly regulated. For example, in people, too much plasticity at the wrong time is linked to brain disorders such as epilepsy and Alzheimer's disease. Additionally, reduced levels of the two neuroplasticity controlling proteins we identified are linked to increased susceptibility to autism and schizophrenia. Similarly, in our fruit flies, removing the cellular brakes on plasticity permanently impaired their crawling behavior. While fruit flies are of course different from humans, their brains work in very similar ways to the human brain and can offer valuable insight. One obvious benefit of discovering the effect of these proteins is the potential to treat some neurological diseases. But since a neuron's flexibility is closely tied to learning and memory, in theory, researchers might be able to boost plasticity in a controlled way to enhance cognition in adults. This could, for example, allow people to more easily learn a new language or musical instrument. In this image showing a developing fruit fly brain on the right and the attached nerve cord on the left, the astrocytes are labeled in different colors showing their wide distribution among neurons. How we did the work. My colleagues and I focused our experiments on a specific type of neurons called motor neurons. These control movements like crawling and flying in fruit flies. To figure out how astrocytes controlled neuroplasticity, we used genetic tools to turn off specific proteins in the astrocytes one by one and then measured the effect on motor neuron structure. We found that astrocytes and motor neurons communicate with one another using a specific pair of proteins called neurologens and neurexins. These proteins essentially function as an off button for motor neuron plasticity. What still isn't known. My team discovered that two proteins can control neuroplasticity, but we don't know how these cues from astrocytes cause neurons to lose their ability to change. Additionally, researchers still know very little about why neuroplasticity is so strong in younger animals and relatively weak in adulthood. In our study, we showed that prolonging plasticity beyond development can sometimes be harmful to behavior, but we don't yet know why that is, either. What's next? I want to explore why longer periods of neuroplasticity can be harmful. Fruit flies are great study organisms for this research because it is very easy to modify the neural connections in their brains. In my team's next project, we hope to determine how changes in neuroplasticity during development can lead to long-term changes in behavior. There is so much more work to be done, but our research is a first step toward treatments that use astrocytes to influence how neurons change in the mature brain. If researchers can understand the basic mechanisms that control neuroplasticity, they will be one step closer to developing therapies to treat a variety of neurological disorders. Sarah DiGenova Ackerman, University of Oregon.